Good morning, students. This is your teacher Neela Mehlawat, and we'll today start with a new topic, that is tRNA. Dear students, I hope you're enjoying studies at home and you're keeping yourself very safe. You're maintaining social distancing and following all the things to keep yourselves and your family safe from COVID-19. Okay, dear students, but don't forget that your studies are very much important. So they should not be ignored at all. Okay, dear students, so keep your books and notebooks ready. We'll start with the topic, that is tRNA. It's a very small topic, but it's a very important one. Okay, dear students, I hope you have no problems regarding the transcription translation. And yesterday we have talked about the mutations, isn't it? The different kinds of mutations like point mutations and frame shift mutations. Okay, so let's start with the tRNA molecule. tRNA is also called as the adapter molecule. tRNA means transfer RNA. tRNA means transfer RNA. Okay, so tRNA, which is called transfer RNA, it reads the codes which are present on the mRNA. Now, dear students, if you remember, we have talked about the DNA, which is a double helical structure. So DNA will form mRNA, and this process is called transcription, right? And this mRNA has the genetic information in the form of codes, in the form of codons, right? And this mRNA, the information which is present in mRNA, will be read by the tRNA. So tRNA will read the information which is there in mRNA, and it will help in the production of amino acids. Okay, dear students, it will help in the production of amino acids. The tRNA is also called as soluble RNA, right? This is also called as soluble RNA, right? And each tRNA has you can say, we know that there are 64 codons, isn't it, students? There are 64 codons, and 61 they code for the amino acids, 3 are stop codons. So for each amino acid there is a specific tRNA, right? There is, for each amino acid there is a specific tRNA, right? And one amino acid can be coded by more than one codon, right? We have talked about proline, isn't it? I hope you remember the examples. So there are in total 20 amino acids, and for each amino acid there is a separate tRNA molecule, right? So tRNA reads the information which is there on the mRNA, and this tRNA is also called as soluble RNA, and there are separate tRNAs for each amino acid. Now this RNA, tRNA, is a single stranded RNA, and it has a clover shaped leaf, right? It has a clover shaped leaf structure, 2-dimensional, and an inverted L shaped 3-dimension. Okay, there is a part which is inverted L shaped, and there is one part which is clover leaf shaped, right? There are 4 double-stranded regions formed by the hydrogen bonding between the bases, that is loop number 3. 3 loops, okay? There are 3 loops. The first loop is called the anticodon loop, right? Anticodon loop means the loop which will read the information present on mRNA. Just now we've spoken about that mRNA has the information for the amino acids. Okay, dear students, now this information will be read by the anticodon loop which is present on tRNA. Second is called the amino acid acceptor end, means this is the end where amino acid will bind. And third is the initiated tRNA. This will start the process, and it has UAC anticodon. If you remember, in mRNA, AUG is an initiation codon. याद है आपको? So AUG is an initiation codon. So the complementary sequence is UAC. That will be present on the initiated tRNA, right? On tRNA, there is no stop codon. right? On tRNA there is no stop codon, right? So this is exactly the structure of the tRNA. Okay, this is the site where ribosome will bind. This is the site where enzymes will bind. This is the site where amino acid will bind. And this loop is called as anticodon loop. And this loop will read the information which is present on mRNA. Okay, this loop will read the information which is present on the mRNA. Clear, everyone? So basically four sites it has. Ribosome, to which the ribosome will attach. Here the amino acids will attach. Anticodons, which will read the information present on the mRNA, and this is the enzyme site. Right, everyone? So it has 4 basic parts. And the structure you see, it is a clover shaped structure. Okay. Now coming to the little bit introduction of translation. We know that DNA will form the RNA. And this RNA will form the amino acids. The process of forming the amino acids from RNA, mRNA, is called translation. Okay, the process of formation of amino acids from mRNA is called the translation process. Now these amino acids then will join together to form a polypeptide. Okay, the number of amino acids will be produced, and these amino acids, they will be joined together to form a polypeptide. Okay, so a long chain of amino acids will be formed, and it is called polymerization of amino acids. Okay, this is called polymerization of amino acids. Now what are the various things that are required for the protein synthesis? So basically we will have 3 things. One is the RNA. First of all, we will require mRNA, because mRNA will have the information, and this information will be read by tRNA, and the work will be done by the rRNA. Secondly, we require ribosomes, because ribosomes, they are the protein factories. I hope you remember in class 9th we have talked about the ribosomes. Ribosomes, they are present on the endoplasmic reticulum. I hope you remember. So they are present on the endoplasmic reticulum, and they are referred to as protein factories because they help in the production of proteins. And the third is the various enzymes. The enzymes which will help in the formation of amino acids. So during this process, 3 things are must. First is RNA, second is ribosomes, and third is enzymes. Let's talk about the RNA. Now mRNA will act as a template. Okay, mRNA will act as a template from which the information will be read by the tRNA. Right, everyone? So this region has some untranslated regions which are called UTRs in front of the start codon. तो देखिए, अगर ये हमारा mRNA है, इस mRNA पर कुछ UTR सीक्वेंसेस मौजूद होती हैं. सो सपोज़ यहाँ हमारा AUG कोडोन मौजूद है, तो ये AUG कोडोन से पहले यहाँ पर मौजूद होती है. ठीक है, एंड दे हेल्प इन द ट्रांसलेशन प्रोसेस. सेकेंडली, tRNA will read this code. Okay, this information which is present on the mRNA, it will be read by the tRNA, and the corresponding amino acid will be produced, and then rRNA will help in addition of the various amino acids. Next, the ribosomes. As I told you, ribosomes, they help in the protein synthesis. Okay, they help in the protein synthesis. So there are approximately 80 different proteins which are being made in our body. So they have structural RNAs, and different 80 proteins will be made. Now our ribosomes, they basically have two units. They are made up of 2 units. One is a large, another is small. You can see in the picture, this is a bigger unit and this one is a smaller unit. So our ribosome is basically composed of 2 units, large and the small. Okay, and when they are separate, they are inactive. ये दोनों units जब अलग अलग होती हैं तो inactive होती हैं. They are active only when they are together. Okay, so when they become active, the small unit and larger units, they will bind together to each other, and only then the process of translation can start. The larger unit has two sides. One is A and P. Okay, one is A and P. So to both these sides, they are very, very important. Okay, the two sides it has, one is A, another is P, and that is very important. A is for amino acid, P is for protein. Now ribosomes in case of bacteria. These ribosomes, they act as enzymes and they are also called as ribozymes. Okay, in case of bacteria, ribozymes, they will act as enzymes and they are referred to as ribozymes, and they will help in the formation of, or they will help in joining the amino acids together. See, these are two amino acids, so they will be joined together by the bond which is called the peptide bond, and this bond will be formed by the ribosome. So ribosomes, they will help in joining of the amino acids together. Okay, dear students. So how many units the ribosome has? It has two units. One is small, another is big. And when the ribosome will be active, when both these units they are together, then the ribosome will be active, and ribosome is called as the protein factory of the cell, right? It is called as protein factory of the cell. Okay, dear students. Okay, we'll take the rest of the process in the next class, in the next video. Okay, this was just an initial introduction to translation. Okay, everyone. So let's come to your homework. First of all, make notes of this topic. So beautifully you will draw, you will write, you will complete your work and underline the important points with blue sketch pen, right? And read the page number 114 of your NCERT book. Dear students, your NCERT is Bible for you. So go through each and every line which is there, right? I try to cover your NCERT in the videos itself. So you must read the page number 114, and if there is any doubt, please let me know that. Okay? So that's all. Bye everyone.